Hey, hello everyone, and welcome to Concordia University's Fourth Space. Thank you for joining us today for the next installment in our ongoing series, taking a closer look behind closed doors and into spaces you might not know about at Concordia. Today, Fourth Space takes a closer look at the Climatology, Hydrology, and Paleo-Environmental Lab, CHAPEL for short. With that, it is now my pleasure to pass the mic to Alex Pache, a doctoral candidate in the Department of Geography, Planning, and Environment. Alex, welcome.
Hey, everybody.
Where are we, Alex?
We're on the 12th floor of the Hall building in the Geography department, and this is our lab, CHAPEL, the Climatology, Hydrology, and Paleo-Environmental Lab. Let me show you around. Come on in. So in this lab, we study environmental change, basically, how it's changing in the future, how it's changed in the past. We use a bunch of different methods. I use tree rings, so I'll be focusing on that today, but we use a bunch of different proxies, they're called. So one of the main things that happens in the lab is we actually get mud samples from the bottom of a bunch of lakes all over Eastern North America. And then in those mud samples, we're able to date them with carbon dating.
Alex, I want to take a look inside this fridge.
Oh yeah, sorry, sir.
Tell me what we're seeing here.
So this is a mud sample from the Ottawa River Valley. We're looking at flooding with that one. Honestly, not sure what all these vials are. This is not my expertise, but basically, there's different things we could do. One of the main things that happens is we go and look at how pollen has changed over time. So some of these vials have pollen in them from different parts of the core. And so some of these cores could be thousands of years old. So we could see how the forest changed over time based on the pollen and then model the climate. And then there's also a bunch of other things you can see in there, like charcoal. I don't know if some of these are charcoal, but you can see how the charcoal changes over time, which tells us about forest fires. You can see how different chemicals change, which tell us about hurricanes if we sample along the coast, which is one of our PhD student Antoine Lachance's project. And yeah, so that's what a lot of people in our lab do. So they sit at these microscopes and they will count pollen grains very slowly over time. It's a bit tedious, but that's part of the job.
I can already tell that we're going to have a lot of fun things to look at and explore here, Alex. So maybe we'll just let the camera go around for a minute and see what we can see, and then we'll ask you some questions about it. How does that sound?
That sounds great. This is a pollen counting corner over here. So we have to do a bunch of stuff to the mud to extract the pollen itself. There's chemicals involved, and so we have a fume hood in the corner to get the air out from the chemicals. Then over here we have a bunch of my samples of wood from underwater, which I'll talk about a bit later. And there's way more wood than this. There's a room stuffed with a bunch of wood. The more wood we could get, the better to understand how the environment has changed, because a lot of these trees are sensitive to climate, for example. So a year that it's really dry, the rings will be thin, and when it's wet, they'll be thick, and then we could use those rings to understand the past. Because some of these trees were started growing like 800 years ago, so we go pretty far into the past here.
These are all your tree rings.
Yes, these are all trees we've sampled we have over 1,000 trees sampled over time. And these are specifically from underwater. I could get into this right away. So we have a few different types of sample types, but these are cedar trees, which is the oldest type of tree in Eastern North America. They could live over 1,000 years. That's pretty rare. None of ours are quite that old, but what we do here is we go to a lake and we dive underwater, get the trees that are underwater, and we're able to pull them out. And they decompose a lot slower underwater because of the bacteria that decomposes them on land isn't there because of the lack of oxygen. And so the trees are preserved really long, and we go really far back in time. And the farther we get back in time, the more context we can get for the changes in climate and environment we're seeing today. And that's one of the types. We really got a lot because you want to have as good as a representation as possible of the tree ring growth in an area. And the more you have, the more you can average out other things that affect the trees, whether it be nearby trees falling down, or insect epidemics, or fires, or whatever it could be. We want to get the climate signal, so we really take as many trees as possible, because the thing that affects all the trees is climate, whereas all these other things can affect more individual trees.
So are you looking primarily at Quebec, or are you traveling around?
Yeah. For my projects, it's all in Quebec. We have our most southern areas in Charlevoix, which is around like an hour north of Quebec City, an hour or two north of Quebec City, and then we go as far north as Charlevoix, which is 14 hours north of Montreal.
Are the only kinds of samples that you take from underwater?
No, not at all. So here, if you want to come around, we don't kill any trees in our lab for multiple reasons, but also we often work in parks, and so we will sample trees when they're alive and get these. And I could show you how that's done, but basically we're able to sample the trees without hurting them because the hole fills up with resin, and that gives us dated tree rings, because the ring right before the bark is the current year, and then we can just count back in time. And there's a pattern of thin and wide rings, and that's what we're interested in, because that means something's affecting them. So we could, I'll let you zoom in on that. And yeah, so every sample, we put the site, we put the species, the sample number, whoever sampled it, so that way if somebody gets a really old tree, they could get the credit for it. And then of course the date. And this one is full of glue. When I first started, I was putting an excessive amount of glue to make sure I don't lose any samples when we go and sand them, because we have to sand them down with a bunch of different grits of sandpaper to really get it nice and smooth. So when we scan it, it pops up nice and pretty.
Great.
To show all these samples we have up here. So this is everything that we have that's like an actual, what we call a biscuit of wood, is dead. It was dead. We don't kill trees. And the way we can actually date these trees, even though they were dead, because we don't know when they died, is by matching the patterns of the dead trees with the ones nearby that are live. So for live trees, in the samples I was showing before, so these guys, we use a borer, an increment borer. So it's this handy tool that to go into the tree. So it's hollow on the inside, and get that sample like this from the tree without damaging it. And then we're able to get the rings for that specific tree and match them to the other ones. So something that's interesting, some of my sites are from beside a lake in the boreal forest, but then others are on alpine areas up north on the tops of mountains. So we're trying to see how climate has changed up there, how the environment has changed. And a really interesting thing is, so this is a spruce that's around 80 years old that you find in the valley. And then this is a spruce as well, found at the top of the mountain that's almost 200 years old. So you could see both difference in color, difference in size, even though this one is way older than this one. And that's mainly because of how stressful the conditions are at the top of the mountain. And another interesting thing about this guy is the center, what you would consider the center of a tree, is the oldest part, is actually all the way at the bottom here. And that's because of a thing called reaction wood. So here we have a nice little cedar that demonstrates perfectly reaction wood. Reaction wood is when a tree falls over or has some sort of force against it, it will put on thicker wood where it fell over. So if you look at this one, all this darker wood here, it had fallen over and was trying to bring itself back up for a couple decades, really, and then it succeeded. Then we have normal rings for quite a long time. And then in 1989, there must have been a huge storm or something like that that knocked down the tree. And so when we found it, it was laying down like this. And for 21 years, until 2010, it put on really thick wood to try to bring itself back up, only to die in 2010, so it didn't succeed. But that's an interesting example of reaction wood. I can actually show you on the microscope what that looks like. We have a beautiful microscope here. And every year a tree puts on a ring. So you can see that pretty clearly with this microscope. It gets darker at the end of the year. Just for structural reasons, it puts on thicker, denser wood. So every time there's that dark point, you could see the end of the year. Now, when that reaction wood starts, which is thicker and stronger, to try and rebalance the tree so that it's upright, you can see how much that changes the color. So the whole ring sort of becomes dark. And then we could zoom in and you could actually see the cells themselves. Whoop, she's a bit damaged because this is a sample I bring around a lot. But yeah, you can see the individual cells there. So when I have a hard time, sometimes trees are so stressed that they hardly put on a ring in a year. And then I put it under the microscope to see, sometimes they're only a couple cells thick. And so that can mess me up when I'm trying to put a date onto the rings. We do a lot of field work in this lab. So we go to pretty remote places, whether it be at the top of mountains or just forests that are untouched in remote regions. And so once we scan a sample, it looks something like this. Let me just fit it to the screen. So we have a high quality sample scan here, and we semi-automatically measure them. So the program could tell just with the contrast of each new ring that there's a ring there. So we just put a line, and then
Alex, it's looking at contrast. It's not a machine learning type pattern recognition.
No, it'd be really nice if somebody would invent that. Maybe I could figure it out because it would save so much time. But some of them are this is a pretty simple one, but some of them, especially in my alpine areas, are really complicated because of how stressful it is and how warped all the rings are and how much reaction wood there is. But this is a nice and simple one. So basically it semi-automatically measures it. Every one of those green spots is a ring. I could zoom in if it's, of course it's lagging. Let's get real close. So you can see how high quality the scan is, and that helps it detect the rings properly. So it automatically puts a ring at each of those. Of course, I'm going to have to go and fix it. Like there's a ring right here that's not supposed to be there. I could get rid of it. And that ends up, it's not cooperating. Okay, there. Then we end up with what we call a chronology. So this red line here, it's just how thick the ring is each year. So you can see it varies. It's not the same every year. And that's what you want, because that means it's sensitive to something, meaning something is affecting the ring. So we do that for hundreds of trees, and then we average them together. So you could look over, I'll bring this over. So these, for my underwater tree site, these are all the different trees. So those, for each of those, it has a chronology of how thick the ring is every year. You can see there's a spot where there's hardly any trees, so something happened at that point. And then what we do with that is we average them all together, and we get a chronology for that site. So that's the average growth of every year at that site. And so that's our goal, is to get a nice long chronology, because then from that we can build different things like a climate reconstruction. And then with that chronology, we are able to go and take, let's say, the precipitation data at a nearby weather station. We'll see that they're actually really connected. So again, when it's really wet, the trees will grow big. That year, when it's dry, they will grow thin. And then we model that relationship for when we have both tree rings and weather records. So let's say in some areas it's like they only started recording in the 1950s, so we have like 70 years of weather records, but then we have 800 years of tree rings. So we can model the overlapping period and put that model on the tree rings from before we had weather data, and that gives us a climate reconstruction. And that's one of the many different things we could do with tree rings.
Alex, one thing I always like to ask is how did you get into this type of research? I know you've been doing similar, like it started in your master's, you're in your PhD now, but how did you get into this?
I got lucky.
You got lucky.
Yeah, I got lucky. I really love field work and going out there and trying to understand the environment. And there was a new professor, Janine Marie St. Jacques, in the department, and I had the right skills and the right motivation to go and do difficult hikes and get difficult tree samples. And then we got along, worked well together, and next thing you know, I was doing a master's with her after doing a little research assistantship, and then a PhD, and now here I am, final year of my PhD, just wrapping up 5 different sites from all over Quebec where we're trying to understand how the environment changed. And it's coming along well. There's a lot of interesting results that I'll be able to share pretty soon.
Alex, right now I can see behind you all the results of all that field work.
Yeah.
It's amazing. Thank you so much for showing us around today and just getting a closer look into your process and what you're working on here in the lab. I think we're going to wrap up the live stream from here, but I'll just do the outro here on the mic, Jack, just to say that we're going to be closing up the Zoom and the live stream, but just remember, this remains available on our YouTube channel if you'd like to revisit it, as you should. And you can check out more of Alex's work just through the Public Scholars website. So search Alex Pache on the Concordia.ca website. And that's it from us.
Thank you.
Thanks,
You're
Alex.
welcome.